Hello, everyone, and welcome to our chat room with Dr. Bliley, who's an expert in 3D bioprinting. Thank you so much for your presence, and we're hoping you're enjoying our event till now. Boa tarde a todos, sejam bem-vindos ao nosso bate-papo com a doutora Bliley. Muito obrigada pela presença de todos vocês. Esperamos que estejam gostando do nosso evento até aqui. I would also like to give a special welcome to Dr. Bliley. It is so nice to finally meet you. Thank you so much for accepting our invitation. It's truly an honor to be here with you discussing such an interesting topic. I would like to thank you again in the behalf of our committee team for taking the time to be here answering these questions. So before we get started, is there anything you'd like to say?
Just thank you so much for having me as well. I'm really excited to be a part of this event, and I'm excited to answer any questions you have. Thanks.
All right, so the first question we have is from Alice. She says, good morning. Thank you very much for the lecture. I found the topic very interesting, and I would like to know if you have tried any other type of printing besides collagen type 1 for the heart.
So we've actually printed a lot of different hydrogel materials. We have done a lot of work specifically in type 1 collagen, but we have printed mixes of like Matrigel, type 3 collagen, as well as alginate, fibrin, PDMS, which is, an inert material that's not normally found in the body, but also is helpful to cells. And we've also printed many different cell types. So I didn't really go into that. We printed cardiomyocytes or heart muscle cells, as well as endothelial cells. So a lot of different materials.
Nice, thank you. I would just like to make a correction. The first question was from Agnes. We have another question here from Talita. She says, how long does it take to develop a 3D bioprinted like these? Excellent lecture, thanks.
So thanks for the question. It really depends on the size of what you're trying to print and kind of the resolution that you would like to print. So for us, you can see a lot of our prints are very high resolution, and we're trying to capture the very small features. So something like a full adult human heart, that alginate print that I showed you, that took like 2 days to print. However, there is some, leeway with how much resolution you need for printing. We probably don't need to print all the very tiny parts of structures, and we can probably get away with lower printing resolution in certain areas. We've also worked a lot on increasing our printing speed, so that requires, transforming our hardware so we're actually capable to print at those fast speeds with high fidelity. And so it really depends on a lot of things, but the larger whole organ prints, at least the heart print, was 2 days.
Thank you, Dr. Bliley. Okay, so we have another question here from Andre. He says, what are the current challenges in developing processing, monitoring, and validation techniques for patient-specific applications of bioprinted materials?
So there are definitely a lot of challenges. One you can think about, especially if we're looking at patient-specific implants, it's, taking either an image from a patient and then creating a patient-specific model. The first challenge is actually validating that does fit with the patient, is exactly what we intended to print. And that's usually done by process term gauging. And we've done a lot of that in our lab. We have pretty accurate reproducibility among our prints, plus or minus 20 microns, depending on how the size of our needle that we're actually trying to use to actually print our structure, which ultimately determines the resolution of the final print. But that's one challenge. And the other challenge is making sure that it actually functions like it should. So with certain like acellular parts, like for example, a valve, we do, sort of cyclic valve testing to see, does it open reliably and close reliably under certain pressures? Also, when does it start to fail? These are all really important questions that we're working on, but it takes time to actually develop a large data set to actually be able to put this into patients. And then the other thing is when it comes to cells, especially heart muscle, we need to make sure that it's contracting with the pressure and generating the pressures that it needs to actually pump blood. This in general is a challenge for the field because the cells that we get from induced pluripotent stem cells or embryonic stem cells are very immature. They're more like fetal heart muscle. So we're actually kind of leveraging some of the strategies that other investigators have looked at trying to mature these cells so that we can actually have them generate the contractile output that we would like them to in order to eventually be able to implant them in the body someday.
Yeah, I'm sure it's an ongoing complex procedure. But we have another question here from Carla. She says, thank you very much for such an interesting presentation, Dr. Bliley. Are these bioprinted valves, tubes, stable enough for a surgery process?
Yeah, so we've actually been working a lot on that. We have implanted some of these into animal models. The valves we're still working on, but if you saw that video, the video that I showed in the lecture, we're able to get them to open and close under physiologically relevant pressures. But we have been able to implant our tubes into animal models at least. One challenge is just trying to, I'm sure as you imagine, they're soft, right? So strengthening certain features of the actual tube that we will be suturing, and we either do that through 3D printing directly by creating like a thicker area, which is able to be sutured into, or locally cross-linking the area just so we can have like a nice area to actually suture into. But we have been able to keep them in animals for a prolonged period of time. And by that, I mean up to 3 months so far. So.
Wow, that is fascinating work. Well, we have another question from Lydia. She says, how can you monitor the time of life of these tissues? You just kind of spoke about that.
Yeah, so I guess the question's for in vivo, unless it's more of an in vitro question. So for in vitro, it's actually, so these heart muscle tissues, like I'm talking about, they are immature. So in vitro, we actually will see them contracting on their own. So it's pretty easy to monitor if they are contracting. That's kind of like a yes or no. But when we implant them in vivo, we rely on things like ultrasound to actually look at the tissue itself. And then also taking the tissue out at certain time points and then analyzing it back in our laboratory. So for that, we would also look at contractile output. We would look at calcium signal across the tissue to look at the conduction of the tissue, as well as some assays like looking just at live and dead cells and seeing how they survive.
Very interesting. Okay. We have another question from Alessandra. She says, hello, I really liked your presentation. I would like to ask, when using the fresh method, how would you make sure that when printing, the needle doesn't drag the soft scaffold along with the gelatin? Is it related only to the texture of the gelatin medium, or the size of the needle also plays a role?
So I think I am understanding what this question is asking. So when we're printing, we're actually printing layer by layer. So presumably if you have an accurate layer deposition, you should actually be moving above that layer and printing onto the next layer. We really don't have any issues with dragging of our actual printed material. And part of that is we're actually depositing a fluid material and it's gelling in the bath. And then it moves up one layer and then starts depositing on the next layer. So we kind of avoid actually running into other layers when we're printing.
Right. Okay. Thank you, Dr. Bliley. Many questions here for you. The next one is from Andre. He says, what are some potential ethical considerations or challenges associated with 3D bioprinting of tissues and organs?
So there are many. I'm sure the thing that comes to my mind initially is cells. So where do we get the cells from to actually generate these tissues and organs? And we focused a lot on induced pluripotent stem cells because we can make them patient specific. But there are some ethical considerations with using stem cells in general. And then the other situation that I think about that comes to mind is if we're able to print organs, which I hope that's what we're working on. We hope we can actually do that someday. Who is going to benefit from that? And then how are we actually going to bring this technology to people? Because right now, actually making the number of cells required to print a whole organ is prohibitively expensive. And actually culturing this tissue to make it have the contractile output that we would like of a whole heart is also very expensive. So we kind of have to think about how, when we develop this technology, how we can make it more equitable for everyone and also reduce the cost so it actually becomes a technology that people will be able to utilize someday.
Yes, very complex as well. Thank you, Dr. Bliley. The next question is from Nadia Nascimento. She says, thank you for your lecture. I would like to know which support material would you use for the decellularized amniotic membrane bioink?
So when we've used decellularized bioinks before, we've actually just traditionally utilized a HEPES or strong neutralizing buffer supplemented support bath. And so this is kind of what we've also used with collagen. So basically when it gels in the support bath, it's because of the actual neutralization of the pH. So that's what I would suggest trying out first. We do know that some of the decellularized materials do tend to work a little bit better when you mix them with another ink, so like a stronger either collagen ink, but we also have printed decellularized materials on their own, but I just wanted to mention that it could be super soft and you might consider mixing it with another ink.
Okay, thank you. The next question is from Dr. Luciano. He says, thank you for your presentation. And the question is, what are the challenges faced when using traditional micro-extrusion-based 3D bioprinting approaches to print soft tissues and organs? And how does the free-form reversible embedding of the suspending hydrogels fresh method overcome all these challenges? I'm sorry, I hope you understood the question.
No, that's great. So I think the thing that Fresh really brings to the table is the fact that we're able to reproduce these very complex structures, not only with materials, but with cells, which is very difficult to do with soft materials. So a lot of the field has relied on strengthening these materials either through UV cross-linking as it's being printed or using some sort of cross-linker during the actual printing process. And we actually don't do that. We can actually print the materials and cells directly because we have the support bath that supports it while it's actually gelling. And then we can actually remove it. So we're able to reproduce very complex prints that are very difficult to produce from soft materials in general, which is very applicable to printing tissues and organs.
Thank you. I'm sure this answer will help us in the lab over here in Brazil. The next question is from Sibeli. She says, is it possible to associate growth factors in the biomaterial used in the bioprinter to stimulate cell differentiation?
Yeah, so we actually have done some work with that. So we've done some growth factors, specifically BMPs for bone differentiation. We've also looked at VEGF for like vascular infiltration and vascularization of constructs. So it's definitely possible. I would say we're just kind of scratching the surface with that right now, but we do have some preliminary work with the BMP and as well with the VEGF that it actually does stimulate those processes.
Great. Thank you, Dr. Bliley. Many questions. The next one is from Luana. She says, how does the cellular differentiation work to produce the tissue?
So I'm not exactly sure, but I'm not sure if it's the cellular differentiation. So normally we actually differentiate the cells in either suspension culture into cardiomyocytes or on a 2D surface. And then we actually have cardiomyocytes and then we print them directly. There are a number of groups that actually do differentiate within the bioprint itself, and they've shown that that's successful. So those groups actually print stem cells and then they differentiate them, usually through just adding factors to the surrounding medium. And there's a lot of good work from Brenda Ogle's group, as well as Mark Schuyler Scott's group, and they've shown that with cardiomyocytes, as well as some neural cells as well.
Great, thank you. The next question is from Andre. He says, how do you see as the future direction for research and development in the field of 3D bioprinting of the heart muscle tissues and other tissues as well?
Yeah, so this is a great question. We have really kind of focused on first trying to print these acellular constructs so that are functional, so like for instance tubes or valves, and we really think that this is the pathway forward if we're able to print these patient-specific implants that can be implanted and then potentially approved at least by the FDA in the USA or elsewhere. The other thing that and so that's kind of like our short-term goal, I would say. And then in the meantime, we're also focused on creating these cellular constructs and at first assessing them in vitro to make sure that they are actually functioning like they should. So for instance, they respond to drugs like they should. They respond to electrical stimulation. They secrete the appropriate growth factors, all of those things, or they have the contractile performance that we want. So a lot of that work is being done in vitro, and then we're moving to in vivo animal studies now. But I think that the path forward is actually first starting with the acellular patient implants, and then moving forward with these in vitro studies. And then once we have the approval for acellular implants, then actually moving towards trying to implant these contractile cardiac tissues. I think first there'll be simple sheets or tube structures, and then more advanced structures that maybe at first will only assist with the pumping of the heart, like not exactly removing the entire heart, but maybe just having a simplified contractile pump that can help assist with the pumping capacity of the heart.
Right. Very fascinating. I'm very excited for future studies. Next question is from Alessandra. She says, have you tried any cartilage biomimetic with this method, like for the ears, nose transplants?
So we've definitely printed ears and noses out of acellular materials. We haven't tried to actually print any cartilage cells before, either like stem cells and then differentiate them later. But that would definitely be something very interesting to look at in the future. And there are some companies and also labs that are also looking at that as well. We just haven't specifically focused on that.
Right. Thank you so much, Dr. Bliley, for answering all these questions. I'm sure everyone is very content with your responses and your presentation. It was great. I would like to read some of the comments that we have complimenting your work. Tatiana said, good afternoon. It's very interesting, smiley face. Elizabeth says, good afternoon. It was an extraordinary presentation. I agree with her. Andrea says, thank you for your presentation. Ciro says, good afternoon. It was an amazing presentation. So as you can see, we all enjoyed it very much. It's been such a pleasure. While I was reading that, more questions came along. Would you mind answering them?
Yeah, of course.
Okay. Lincoln said, hello, Dr. Jacqueline. Firstly, thank you for your presentation. It was awesome. In my thesis, I'm trying to develop 3D valvular organoids to study the COPD with more accuracy compared to the traditional models available. Have you ever tried to bioprint pulmonary cells in the bioprinter? Kind regards.
So I personally have not. We do work with some groups who are trying, not for COPD specifically, but are trying to print pulmonary cells. I have no experience with that, so I can't really answer this question. But if you would like to send me an email, I'd be happy to chat with any additional information that I can provide.
I'm sure you have your hands full with your work now. We have another question from Alessandra. She is very interested in the topic. She says, what would be the implications of applying soft electronics to the printed structures to add function or movement to the biomimetic?
So that's actually a pretty cool idea. So we are collaborating with a group that is very interested, and we also print PDMS, which is a typical soft elastomeric substrate. And yes, you could definitely do that. I think we're in the very early stages of actually trying to do that, but you can imagine either stimulating or recording from certain regions. That's something we're very interested in. One, because it will tell us more about the function of our tissue. Like I said, these cells are very immature. So if we can monitor them over a long time and kind of see how they change, we might be better able to understand the maturation of these tissues over time.
Okay, great. Another question from Ulysses. He says, thank you for sharing your lecture. It was excellent. Are there spin-offs from these developments?
So I'm guessing that means spin-off companies, but so we actually do have a startup that started out of our lab, Fluid Form 3D. It's located just outside of Boston, Massachusetts, and they are doing very exciting things. So yes, there is stuff that's happening. They're more so focused on the translation of what we're doing. We're kind of focused more on like building the tissues, which is really fun. But yeah, there is a company that has started from this work.
Okay, thank you so much, Dr. Bliley. So it seems that we have ended the question section. I don't believe there are any more on here. Many compliments and comments for your presentation. Dr. Bliley, I would like to thank you so much for taking your time for being here with us today. It was excellent, as everyone agreed so. Yeah, thank you so much for everything, and I hope to see you soon again in other studies. It was a pleasure in the name of our committee.
Thank you so much for having me. I really enjoyed it. I'd be happy to answer any other questions via email if anyone would like to email me. Thank you.
Yes, sure, of course. We will be in contact. It was very nice. We heard great things from Dr. Adam from you, so it was great having you. Thank you so much. Okay, thank you everyone for your time and patience. We have a few minutes before the next presentation. Thank you so much for all the questions, and I will see you guys in the next presentation. Muito obrigado a todos pelas perguntas e pelo tempo aqui conosco. Temos alguns minutinhos até a próxima palestra. Então, nos vemos daqui a pouco. Muito obrigada. Bye, Dr. Bliley. Thanks.
Bye.